Hello everyone, welcome to another series of video presentation. Here we will discuss cells of immune system or the different cells which are associated with immune responses. In the previous video we have already seen that different organs associated with immune mechanisms, where we have seen the primary organs and secondary organs. And the primary organs, where we define the primary organs are the sites where the immune cells develop and undergo maturation, right? And the secondary organs are the sites where the cells interact with the foreign substances or the antigenic substances in order to elicit an immune response. So in both that cases we know that these immune cells are playing a crucial role, right? So we need to talk about immune cells in detail, where these immune cells are found from, or where these immune cells are generated, and how these immune cells are generated, what is the mechanism of immune cell generation. All those things we will see in this part of, or in this series of video presentations, right? So let's move to the content. So we know that this is a kind of introduction, like there are cell mediated immunity or adaptive mechanism. Whatever be the mechanism, the cells are playing crucial role in immune responses. And the major cells involved in immune responses are what we call the WBCs or leukocytes, or in short or otherwise the white blood cells, right? So these are playing the crucial role in the defense responses, right? And where they are found, actually they are found, basically they are found in the body fluids like blood, lymph, and also seen in the immune organs like the secondary organs especially, and also in the tertiary organs like spleen, lymph node, the malt, the mucosal tissues or the cutaneous tissues. All those sites are the location where we can find these immune cells. And there are different types of leukocytes. This may be familiar to you that there are different types of leukocytes available. Based on, we can categorize them based on their morphology, their presence of granules, presence of receptors, and also based on their different functions in the performed in the body, right? So when we talk about the location of synthesis, already we have seen when we talk about the bone marrow that that was the site for the synthesis of blood cells, right? That's true that all blood cells are synthesized in the bone marrow, right? And after synthesis they are released into the body fluids, particularly into the blood, and from the blood they migrate into various other parts including lymph and other secondary organs, and where they interact with the foreign substances, and that is what we call the immune response, that leading to kind of effector mechanisms, and ultimately this foreign substance or the antigenic substances will be eliminated. And we know when we talk about cells and organs, we also have to note that all these cells, organs, and all molecules associated in immune response are having some interpersonal communication, or they are having enough communication between these cells and organs, and this is done through various chemical substances or signaling molecules like cytokines and chemokines, etc. So they are actually playing a crucial role in the regulatory mechanisms of any immune response. So here in this slide we can see the different cells seen in blood, or these are the immune cells, and these cells not only seen in blood. You can see here, you can see all these cells are located in blood, and these are certain cells which are found in lymph and lymph node, and these are certain cells found in tissues, right? So here you can see the B cell, T cell, NK cells. These three are coming under the lymphocyte category, and here you can see neutrophil, eosinophil, basophil. These three are the granulocytes. Then here you can see the platelets, erythrocytes. These are the or otherwise in short, erythrocytes are coming under the RBCs. And these three dendritic cells, mast cells, and macrophages, actually they are found both in blood and tissues, right? They can be present in blood and body fluids, but also found in tissue spaces, right? They are responsible for a very common significant function called phagocytosis. Macrophages are basically involved in that, and mast cells are and dendritic cells are also doing various other functions in the body, right? So lymph nodes are also populated with lymphocytes like B cell, T cell, NK cells, and also the dendritic cells. So these are the different cells seen in immune responses, right. So when we talk about the synthetic process or the process by which the blood cells are synthesized, that event, that phenomenon, that process by which all the blood cells are synthesized is called hematopoiesis. So this occurs in the bone marrow as already stated. So these cells actually originate from a common cell or from a mother cell called stem cell or hematopoietic stem cell, right. So I hope you know what is a stem cell. A stem cell is nothing but a cell, or it's a kind of mother cell from which all the different types of cell could originate, right. That means they are capable of differentiation. They are having two capacities. One is they can propagate themselves, that means they can undergo self renewal, and the second thing is that they can undergo differentiation into different types of cells, right. And within the bone marrow, the number of these HSCs are comparatively very less, like 1 in around 10,000 cells. Very few number of HSCs are commonly found in the bone marrow, right. And yeah, this is already said, self renewing, and they undergo, it's even though they are self renewing, the division is very slow. That means they divide like in 10 to 12 months, once in 12 to 12 months, 10 to 12 months, right. Then yeah, they are multipotent or pluripotent, which means they can differentiate into different types of cells. That means from the same cell 10 or 12 different types of blood cells are originating or differentiating, right. So I am not going much details into the stem cells. You can refer stem cells in detail for the properties like multipotency and pluripotency. So this one, you know, in the previous slide we have seen different cells. So this table shows you the concentration of different blood cells. You know, RBCs are the richest cells, then followed by platelets, then leukocytes, and within leukocytes, you know, neutrophils are the leading cells, followed by lymphocytes, monocytes, and so on, right. This one will give you a more detailed idea about that. You know, lymphocytes are again split into different types like T cells, then there are subtypes of T cells like CD4 and CD8, and also B cells. Their particular concentration in adult and children are given for reference, right. So whenever there is a variation in these concentration, that leads to some kind of disorders. And yeah, this is the image which we can use to explain hematopoiesis, right. Here you can see there is a self renewing hematopoietic stem cell. This is the HSC we are looking for, right. These stem cells are found in the bone marrow, and these stem cell will undergo differentiation into basically into two categories. One is called the lymphoid progenitor, the other one is the myeloid progenitor. This is the process of hematopoiesis by which the blood cells are synthesizing. You can see all those blood cells which we have listed there has been listed here also, like a T cell, B cell, all those cells we can find here also. So their route of synthesis is also shown here, right. So you can see there are two basic lineages, basic progenitor cells coming from the hematopoietic stem cell. One is the lymphoid progenitor, the other one is the myeloid progenitor. So the lymphoid progenitor will further differentiate into the lymphoid cells or the lymphocytes in common, right. And the major lymphocytes or the predominant lymphocytes are T cells and B cells, right. So they are coming from another progenitor cell called T cell progenitor and B cell progenitor, all right. And additionally we have natural killer cells and dendritic cells also coming differentiated from the lymphoid progenitor, all right. And when we talk about the myeloid progenitor, we have a number of cells, a number of progenitors, like all those progenitors for monocytes, neutrophils, eosinophils, basophils, even platelets and erythrocytes. All those are coming from the myeloid progenitor, right. You can see here you can see the megakaryocyte. These are the cells from which the platelets are originating. These platelets are, you know, these are small structures without a nucleus. Actually they are coming from division of these megakaryocytes, and erythrocytes are also coming from the erythroid progenitor cells, right. And here is an important progenitor called the granulocyte monocyte progenitor, which differentiate into, or which can differentiate into neutrophils and also into macrophages. In between you can see monocytes. These monocytes are the type of cells seen in blood basically, and from when they migrate into tissues, they transform into a cell type called macrophage, right. And here you can see the dendritic cells here also. Here in the lymphoid lineage also you can see a dendritic cell, and also in the myeloid lineage, right. Yeah, this one also will give you the same idea of hematopoiesis. Here you can see the lymphocytes, then all other cells, monocytes, neutrophils, all those things are there. So this is the explanation for the hematopoiesis. I think it's already done. We have the myeloid progenitor and lymphoid progenitor, and one thing more to add here is the stromal cells present in the bone marrow will control the hematopoietic process. That means the environment required for the hematopoiesis is provided by the stromal cells, right. Otherwise the hematopoiesis will not be proper. So the differentiation, the route of differentiation, or which type of cell has to be synthesized, all those things are decided or regulated by the stromal cells. And how the stromal cells will do that? It is through interaction of chemical signals called cytokines, right. So which lineage of the cell to be synthesized is decided by the regulatory molecules like cytokines, which are basically produced by the stromal cells on interaction with the stem cells, right. Not only the stromal cells, but T cells and B cells are also associated for the synthesis of these cytokines. Then when we talk about the cytokines, there are two categories of cytokines. One is promoting the hematopoiesis, the other group is inhibiting the hematopoiesis. Here you can see the examples for those which are promoting, like interleukin 4, 6, 7, 11, GM-CSF, EPO, etc., etc. Then the inhibitory cytokines are MIP, then interferon, then TNF, etc. These are the inhibitory cytokines, and this table will give you a detailed idea about that. The different cytokines produced by different cells, how they affect on the stem cell, and which type of cells are either proliferated or inhibited. This table will give you a detailed idea about that. So how do we regulate hematopoiesis? Because regulation is very important. It's because when we talk about blood cells, we are familiar with a common concept that blood cells undergo cell death by a period of around 120 days, right. Is that true or not? It is true, right. It is true because there are certain cells like RBCs given there. Certain cells like erythrocytes will undergo cell death within 120 days. That means around 90 to 120 days are the normal lifespan of erythrocytes. But that's not true for all the blood cells. When you talk about neutrophils, they live only for few days. But at the same time, when we consider certain T cells or certain memory cells, those cells will survive for long years, right. So means that there are different cells with the different lifespan, right. So there should be a kind of regulation for hematopoiesis. That means the system has to check which type of cells are present in which concentration in the blood, and accordingly a signal has to be given to the stem cell, and then the stem cell as per the signal received will synthesize a particular type of cells, and this basically done to maintain the steady state of cells in the circulation, right. So there should be a balance between the number of cells which are undergoing cell death and the number of cells which are newly generated, right. And also already we have seen in the when we discussed about the organs of immune system, when we talk about the spleen, we have seen that the mature blood cells including the RBCs and WBCs, undergo cell death in the spleen by a process there is an elimination process in the spleen. And there is another process also for the elimination of aged cells called apoptosis, right? So this elimination process is called apoptosis. It's a programmed cell elimination process or a programmed cell death mechanism. It's a tightly regulated mechanism so that no harm will be there for the system. There is no harm for the host body by this controlled mechanism. All right. So apoptosis is a topic of another discussion. Right now I am not moving into the process of apoptosis. So this is the last slide for this part of presentation. Here you can see a representation of how the regulation occurs. This is a kind of demonstration. This BCL2 is an example for an activation signal, or the activated B cell will release a signal called BCL2. And BCL2 is a molecule which can prevent apoptosis, right? So that when we have high concentration of BCL2, the B cell will never undergo apoptosis, right? But if the level of BCL2 is less, that can cause the cell to move to apoptosis, right? Apoptosis in the sense they undergo cell death, right? So normally when the activated B cells comes out of the system, the concentration of B cells will be decreasing, and automatically the cell will have a tendency to undergo cell death. But when there is a continuation signal, when there are certain additional signals like additional cytokines, T helper cell, or the antigens, when these things are coming, these are collectively called as continued activating signals. So when we have these signals present, these B cells will undergo differentiation, right? Instead of going to the cell death, they undergo differentiation and they will do their rest of the functions, right? So in that way these cells undergo a kind of regulation. So accordingly, when we have a more number of cells undergoing cell death, then the communication cell signal will go into the hematopoietic stem cell and they will differentiate more in number. So that's about this regulation in short, right? So by that we'll wind up this part of presentation. I hope you understood about hematopoiesis. I'll brief once again. Hematopoiesis is the process by which all the blood cells are synthesized, and the process occurs in the bone marrow from a common progenitor cell called hematopoietic stem cell. They differentiate into two lineages, lymphoid progenitor and myeloid progenitor. The lymphoid progenitor will differentiate into lymphocytes, like including B cell, T cell, NK cells, and the myeloid progenitor will differentiate into all other blood cells, all other leukocytes, right? And these cells, or the process of hematopoiesis, is under tight genetic regulation, right? So thank you for listening.